section, which is talking to Martina. And Martina works for SAMS. She is currently in France, so she has some fantastic weather there at the moment. But Martina has been working in the field of seaweed for the last 10 years, looking at pathogens within the seaweed sector. So without further ado, Martina, last but not least, can we unmute yourself? Sorry, I can't seem to.
I am unmuted, muted,
Perfect.
yes.
Very
I'm not
good.
sure whether you can see my screen already or not.
Yeah, we can.
Okay, brilliant.
Great.
I won't start my video because my computer just crashed during Antoine's presentation, so I'm a bit afraid that it's a bit
no problem.
unstable. Okay, so I'm just going into the presentation mode. Okay, is that fine for everybody? Okay, brilliant. So yeah, good morning, everybody. My name is Martina Strittmatter, and I'm working at the Scottish Association for Marine Science as a postdoctoral research assistant in algal pathology. And yeah, thanks for the invitation to talk to you today. And I will focus on seaweed pathogens. So the seminar really will be divided into 3 parts. In the first part, I will give a brief introduction and some definitions about seaweed diseases. And then in the second part, I will show you some concrete examples of seaweed pathogens that are currently found in seaweed aquaculture. The last part will focus on some tool development and establishment of a database that we are currently setting up in order to gain a better knowledge on algal pathogens. Here at this point, I would also like to introduce and acknowledge our team here at SAMS, which is led by Dr. Claire Garchon, and they all contribute to the various aspects of seaweed diseases. So like any other living organism, algae can be plagued by diseases. Diseases is in a very broad sense, these are any abnormal physiological or developmental conditions that can be caused by abiotic or biotic factors, or even a combination of both. Today I will focus on biotic stressors like pathogens and pest organisms only. So these pathogens and disease-causing organisms of seaweed are of a diverse phylogenetic origin, and they are omnipresent. And here I just show you a few examples of such algal pathogens in the eukaryotic tree of life. So here, for example, we have oomycete pathogens and brown algal pathogens. Then we have fungi that can infect macroalgae. We have green algal endo and epiphytes that can be a problem for algae. Then we have red algal parasites, and for example, here we have phytomyxid pathogens. And of course, macroalgae can also be infected by viruses and by bacteria. So such algal diseases, they are documented in both in wild and cultivated macroalgae. When talking about seaweed pathogens and their impact on aquaculture, I think a very prominent case in this regard is really the Porphyra industry in Asia. So Porphyra sensulato is an important marine crop with an annual market value of around $2 billion US. It is commonly used as a food source, and you can find it in sushi here, for example. And the main producers are Japan, China, and Korea. And here, this just gives you an overview of a Porphyra farm in Korea. So there are several organisms that currently challenge the cultivation of Porphyra. So first of all, we have the real pathogens that infect and kill algae. And here we have 2 oomycete species that are known to infect Porphyra. So first of all, we have Pitium porphyra, which is causing the red rot disease. And then we have Pediopsis porphyra, which causes the Pediopsis blight. Macroscopically, they evoke very similar disease symptoms, and they consist of discolored patches, necrotic zones, and lesions. On the microscopic level, those two can easily be distinguished. So the first pathogen, the Pitium, forms those hyphae, which build a network between the cells of Porphyra, whereas the Pediopsis pathogen is an intracellular pathogen and infects single algal cells. There's also a third pathogen disease, which is affecting Porphyra, and this is called the green spot disease, which is caused by viruses and bacteria. And apart from those pathogens, the Porphyra cultivation is also affected by fouling organisms like, for instance, cyanobacteria and diatoms, which are firmly attached to the surface of the alga. So the impacts of those pathogens on the aquaculture production of algae was recently quantified in a major seaweed farm in Korea. So here I show you a graph, and you can see the incidence rate for the three major diseases: the green spot disease, the red rot disease, and the Pediopsis disease during the cultivation period of 2012 and 2013. The Pediopsis disease was very prominent in this year 2012-2013, and it actually reached an incidence rate of up to 70%. And this disease actually led to an early harvest of Porphyra and meant a shorter growth period. The production loss due to this pathogen was estimated to be around $1.6 million, and this corresponds to around 25% of the annual sale in this cultivation area. In previous years, this local industry had mostly been hampered not by pathogens, but by fouling diatoms, which didn't cause any loss of biomass, but this infestation with diatoms reduced the auction price of the Porphyra crop, because those diatoms that were attached to the surface actually changed the taste and the visual aspect of the Porphyra blade. A second important seaweed sector is the carrageenophyte industry. And here the main carrageenophytes cultivated are Capaphycus and Eucuma species, and main producers here are Indonesia, the Philippines, Malaysia, China, and Tanzania. The eucumatoid cultivation constitutes for the biggest annual production in terms of fresh weight. Here we also have two major diseases and pests that occurred during the Capaphycus and Eucuma farming. So first of all, we have the ice syndrome, and then we have epiphytes. So here in the middle, you can see an image of healthy algae, which occur in different color variants. Then here on the left-hand side, you can see algae that are affected by the ice syndrome, and this causes the characteristic bleaching of the alga. And then here on the right-hand side, you can see algae that are infested by epiphytes, which gives the Capaphycus this hairy aspect. Those epiphytes are known to cause major production losses. In the Philippines, for example, the yearly average loss due to epiphytes is estimated to be around 17%. And then likewise, the annual production of those eucumatoids is greatly varying here in Tanzania and Madagascar due to those epiphyte infestations. Apart from this, I think the carrageenophyte cultivation gives also a very prominent example of germplasm movement and unwanted side effects. So when you see the map here from the late 1970s, Capaphycus has been introduced from the Philippines to various countries within or without its native range. And then after several years of cultivation in those countries, the seaweed became naturalized or even invasive in some of those regions. And moreover, disease outbreaks like the ice and the epiphytes started to appear in some regions. And this is really suspected that the pathogens have been introduced unintentionally via contaminated seed stock. And in addition to this damaging effects for the seaweed aquaculture, those diseases also represent a risk for the local wild seaweed populations. So whereas the previous slides, they showed the impact of pathogens during the stage of biomass production, I will now switch to some pathogens that can impact the seeding stages, so the hatchery stages. So here again, we look at the red alga porphyra, but this time we look at the diploid conchuseli stage, which is used in seeding farms for the preparation of seedling material. So this conchuseli stage of porphyra is microscopic and shell boring. Those shells are cultivated for several months until the material maturates and releases spores, which are then used to seed the new cultivation nets. And like the blade stage that I have just shown you before, also the conchuseli stage is prone to infection. And here we have two prominent examples that have been described in the literature. So here on the left-hand side, we have the white spot disease, which is caused by a fungus. And then here on the right-hand side, we have the yellow spot disease, which is caused by certain bacteria. The common characteristic of those two pathogenic agents is that they cause bleaching and death of the conchuseli stage. And therefore, they can have an impact on the availability of the seeding material. However, the extent of pathogen-related damage at this hatchery stage is currently not fully understood. So here in this slide, I would like to draw your attention to a recently discovered pathogen in Palmaria palmata, which has been discovered from Scotland. So as you probably all know, Palmaria cultivation relies on the production and release of tetraspores from fertile tetrasporophytes. So there were cultivation facilities that have repeatedly reported seeding failure. And then my colleague Yasin Badis actually had the opportunity to analyze such fertile tetrasporophytes and tetraspores. And he found that the tetraspores were actually infected with an intracellular pathogen, which I show here. So here on the left-hand side, we have a healthy Palmaria spore. And then here on the right-hand side, we have Palmaria spores that are infected with this pathogen. So here in this infected material, you can see several pathogenic thalli within the cell. And then little by little, those pathogenic thalli, they expand and really fill up the whole algal spore. And then at the latest stage of infection, the pathogenic thalli develop into sporangia, and then they release pathogen spores into the seawater. And at this stage, the algal spore is completely dead. A molecular barcoding analysis of this infected material revealed a new species of pathogen, which was named Pediopsis palmaria. And this is actually a pathogen closely related to the porphyra pathogen that I have just introduced a few slides earlier. And we can assume that this pathogen contributes, at least to some extent, to the difficulties that are experienced in the Western aquaculture of Palmaria palmata at the moment. So now I would like to introduce you to a more modern approach of pathogen detection. So the pathogen that I've just shown you in the previous slide, it has been discovered by a classical approach of symptom detection, microscopy, and molecular barcoding. Such an approach allows to investigate the life cycle of pathogens and in the investigation of host pathogen interactions, but it is a rather low throughput method. and has some spatial and temporal constraints. So my colleagues have developed a different approach of an in silico data mining of publicly available data sets. So once you have your DNA signature of your pathogen of interest, you can use this signature to detect homologous sequences from large meta-barcoding and meta-genome data sets. And this has been done with the pathogen Pediopsis using a custom pipeline, and this allowed to retrieve 2 sets of informations. Oops. So first of all, GPS coordinates could be extracted from those data sets, and this actually allowed the mapping of the pathogen. So here in this graph, you can see the previously described Pediopsis pathogens here as asterisks. And in circles, you can see the Pediopsis sequences that have been revealed by using this data mining approach. And this really shows you here a global distribution of this Pediopsis pathogen. Secondly, those homologous sequences can be retrieved via this data mining approach, and they can be used for phylogeny. And here, in the case of Pediopsis, this allowed to discover a large unknown diversity of the genus, and probably there are much more new species that still need to be described. So we can assume that at the moment this red algae pathogen is really underdescribed and underestimated. So from those previous slides, we could see that there's a rising concern about macroalgal diseases, specifically in the regard to cultivation. There's a potential risk of pathogen spreading between wild and cultivated species, and there is also the potential risk of pathogen transfer with cultivars between cultivation sites and so on. And despite those risks and rising concerns, the current understanding of those algal pathogens is still very limited, and no baseline is currently available to address questions like diversity, biogeography, host range, and ecology of such algal pathogens. So in order to further and accelerate our current knowledge of those algal pathogens, we have recently set up a participative web portal, which is called My Seaweed Looks Weird. So I would really like to invite you to visit our My Seaweed Looks Weird web portal, which can be accessed here under this address. So with this web portal, we really aim to provide a platform where everybody can report observations of diseases in cultivated and wild algae. And it's currently available in 3 languages, in English, Spanish, and Portuguese. So when we're talking about possible disease symptoms of infected algae, so there are some macroscopic signs that can indicate the presence of putative pathogens in macroalgae. So first of all, there are galls, blisters, and warts, as you can see here. Then we can also distinguish spots, bleaching, discoloration, lesions, holes, and also distorted morphology. So this can all be indicators of some pathogens being present. In addition to simple observative data, we furthermore encourage the submission of material. So such submission can consist of image material, which, for example, documents the unhealthy, unusual looking algae, or we even encourage the submission of physical samples. So my colleague, Dr. Pedro Murua from the Austral University of Chile, he's in charge of the samples coming from Latin America, and myself, I'm in charge of the samples that come from Europe and the rest of the world. So if you are interested in sending samples, I would really recommend to contact us prior to sample shipment, and because we can advise on storage conditions, fixation buffers, and we can also assist in the arrangement of shipments and so on. So once we have received potentially diseased samples, we can envisage several analyses, which largely depend on the fixation and storage conditions of the sample. So in the first instance, we will always perform a microscopy analysis, for example, with specific stains, in order to get first indications of the pathogen structures and host cell reactions and so on. If possible, we would then also try to get barcode sequences of the putative pathogens, but this might sometimes be difficult or even impossible, depending on, for example, the storage conditions of the sample and so on. Additionally, we can also try isolation, cultivation, and reinfection of those putative pathogens. And we will offer an in-kind analysis of the samples that have been submitted via our My Seaweed Looks Weird web portal, and we will, when possible, establish reports of our observations. So our objectives of this web portal are really to encourage the participation and the sample submission on a large scale in order to accelerate the description and identification of such algal diseases worldwide, and also to increase the knowledge on the diversity of such algal pathogens. And last but not least, we aim to make the gathered information available in an open access repository, which is called the Algal Disease Atlas. So this disease atlas is currently in preparation, and it will contain some taxonomic data of pathogens and host algae, as well as images, like for instance, different life cycle stages of the pathogen or microscopic and macroscopic disease symptoms. Furthermore, it will contain some biogeographic data, literature, and protocols. The data that are going into the disease atlas, they originate to a large extent from already published literature, and we aim to also include relevant information from non-English literature. Furthermore, data obtained from the ongoing projects Genealc, Global Seaweed Star, Alga Blister, and from the My Seaweed Looks Weird web portal will also be included in this disease atlas. And we aim to publish a first version of this atlas at the end of this year. And really the overarching goal of this atlas for us is really to establish an informative database that can be useful and consulted by a broad audience, and really to advance the exchange and collaboration in regards to seaweed diseases. And on this note, I finish, and I would like to thank you for your attention.
Thank you, Martina. That was a very interesting presentation. I personally haven't seen any seaweed samples, thankfully, with any of those issues previously. Just one question for you in terms of that service that you mentioned about sending samples. Is that a free service for businesses or people who'd like to send in samples, or is there a cost associated?
No, currently this is a free service, so everybody can really get in contact with us, describe a bit what the problems are, and then we can discuss together whether we would go further for sample analysis and so on. Yeah.
Great. Okay, that's fantastic. Very, very useful to know about that.